So this is a presentation of the software we developed in the lab, which is called Scratch, and which is designed for wound healing assay. So when you run the program, it will open this window. So on the left side, you will have all the commands, and on the right side, you will have all the visualization. So about the menu, each block can be open or closed depending on what you want to see. Or once you want to hide, you can also reorganize them or even create different columns if you prefer. And you can close everything so to only keep the graphs in the window. So let's try for an experiment. So the first step is to import the images. So if I go to import, you go to the folder where you have your images. So your images are just TIFF image in a sequence which has been acquired on your favorite microscope and can be converted in a specific way using a small macro in image. So I open all of them. So opening it, it will directly recognize the pixel size and also recognize the time lag between the images. So all the parameters are directly read from the file. So the first step is to detect the two borders. So for this, I will have 4 parameters, and the two most important ones are the size and the score. So you can decide the value that you want, and you can go to single to analyze the current image. So you can see that you create there an image where you have green on one side and red on the other side. If it's not so clear, you can hide the other window, and so by this way you have just the image you want to work on. So this is a set of parameter which is not so bad, but if you want to explore other possibility, you can go directly to tester. So tester is used to scan different set of parameters for score and distance. So when you launch this add-on, then you will have the current set of parameters. So it was 10 and 0.07, and it will test in a range of plus or minus 2 times the delta, both in distance in horizontal or in score in vertical. So you can decide to center on another value. So let's go on 18.27, and it will move this one in the middle, and now I have all the range around this set of parameters. I can also decide to zoom in terms of distance, because now in terms of distance I'm at plus or minus 8. So if I go to zoom distance, now it will go plus or minus 4. So it's 2 times finer range in horizontal. In the same way for score, you can zoom score to improve the precision of the range of values. So let's go back to this one, which seems to be better. And when I'm happy with the parameters, I can just go back to the main software going with finished. So now it has taken the parameters in the auto detect, and for this image, so I have the parameters as seen before. So now to do it for the whole stack, I just have to click on full, and so it is analyzing each single image. So we can see that some cells are leaving the edges to go in the middle, so that's why they were blue. And now at the end I have almost fully closed wound. So now I have some data. Now let's see more in details about other parameters, and to extract in fact some quantitative data. So now I go to analysis and export, and I go directly to analysis, and then it's scanning each image and optimize the detection of the borders. Now I have a single line and not all inside. I can of course plot the area of side 1, side 2, or the whole. So That's why I have something which is almost linear for the progression of the two fronts and reduction of the whole, of course, and I have the linear fit, which is extracting the mean speed of progression or reduction of the whole. So I have other parameters. So basically one part is all the question about the neighbors. So the idea of neighbors is that I analyze for each pixel of each side how far is the closest pixel of the other side. So by this way I have many data for the statistics, and it helps me to have a better idea about how the edges are evolving with respect to each other. So having statistics, I can extract the distribution, and so I can see on this graph to visualize that at the beginning all the pixels are around 300 microns from each other, and then with time the distance is reduced, and also we can see that I have an increase of the width of the distribution, meaning that the patterns is more and more complicated, and at the end of course it's going to something which is pretty sharp, because it means that the wound is closed. So this is a graphical representation. I can also extract so the mean value, which is the black dots, and if I check the standard deviation, I have an idea how it's closing with time, and how wide is the variation of the distance. On this graph I have three fits, so one which is hyperbolic tangent, one is linear, one is quadratic, and in fact all the parameters are stored in the log window for direct visualization. So the standard deviation of the neighbor's distance is shown here, but it's also shown here, and so by this way you can see some, in some cases you can see some evolution of the standard deviation, which gives some information about how evolutes the fronts. So now I saved, I already saved, sorry, with the analysis an Excel file in the folder where was the teeth images, but I can also export each single graph. So, but this way I will have a snapshot of each file. And what can be done in the program when I'm happy with the parameters is that I can in fact save all the parameters in a slog format, so it's a specific format, and when I save them, I have all the information which are required to redo it at the end. So now if I load this, so I go basically from the first step with all the parameters. Now I can directly go to autocomplete, which will make all the steps in a sequence, so it will be a fast way to have a complete reproduction of the results if necessary. So in this autocomplete, it will make so the full detection, then the analysis, then the export, so the Excel file and the TIFF images, and it will also save the slog file. So this is the software, how it works, and hoping it will help you if you have to use it.